Dear viewers, this video is part 2 in the three-part series on cancer genetics in the Medicine for Anyone YouTube channel. Bladder cancer. Somatic mutations in the FGFR3, PIK3CA, KDM6A, and TP53 genes are common in bladder cancers. Each of these genes play a critical role in regulating gene activity and cell growth, ensuring cells do not grow and divide too rapidly or uncontrollably. It is likely that mutations in these genes disrupt normal gene regulation, contributing to the uncontrolled cell growth that can lead to tumor formation in bladder cancer. Note: FGFR3 gene provides instructions for making a protein called fibroblast growth factor receptor 3. This protein is part of a family of 4 fibroblast growth factor receptors that share similar structures and functions. These proteins play a role in several important cellular processes, including regulation of cell growth and division, that is proliferation, determination of cell type, formation of blood vessels, that is angiogenesis, wound healing, and embryo development. The FGFR3 protein spans the cell membrane so that one end of the protein remains inside the cell and the other end projects from the outer surface of the cell. This positioning of the protein allows it to interact with specific growth factors outside the cell to receive signals that control growth and development. When these growth factors attach to the FGFR3 protein, the protein is turned on, that is activated, which triggers a cascade of chemical reactions inside the cell that instruct the cell to undergo certain changes, such as maturing to take on specialized functions, that is differentiation. Several versions, that is isoforms of the FGFR3 protein, are produced from the FGFR3 gene. The different isoforms are found in various tissues of the body, and they interact with a variety of growth factors. Many isoforms are found in the cells that form bones. The FGFR3 protein regulates bone growth by limiting the formation of bone from cartilage, a process called ossification, particularly in the long bones. One particular isoform of the FGFR3 protein is found in cells that line the surfaces of the body, that is epithelial cells, including the cells that form the outermost layer of skin called the epidermis. Bladder cancer is generally divided into two types: non-muscle invasive bladder cancer, that is NMIBC, and muscle invasive bladder cancer, MIBC, based on wherein the bladder the tumor is located. Approximately 80% of NMIBC tumors have FGFR3 gene mutations. FGFR3 gene mutations change single amino acids in the FGFR3 protein, which appears to overactivate protein signaling. As a result, bladder cells are likely directed to grow and divide abnormally. This uncontrolled cell division leads to the formation of bladder cancer. So, dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Renal carcinoma. The genetic changes are trisomy of chromosome 7, loss of Y chromosome occurring in patients older than 60 years old, and 3. Loss or rearrangement of material in 3p, that is chromosome 3 short arm. Two types of cancer genes: oncogenes and tumor suppressor genes. Oncogenes and tumor suppressor genes exert their effects on tumor growth through their ability to undermine cell phase, influence cell survival, and contribute to genome maintenance. The underlying molecular mechanisms can be extremely complex. While tightly regulated in normal cells, Oncogenes acquire mutations that typically relieve this control and lead to increased activity of the gene products. This activating mutational event occurs in a single allele. Note, an allele is one of two or more versions of DNA sequence, that is a single base or a segment of a base, at a given genomic location. An individual inherits two alleles, one from each parent, for any given genomic location where such variation exists. If the two alleles are the same, the individual is homozygous for that allele. In contrast, the normal function of tumor suppressor genes is usually to restrain cell growth, and this function is lost in cancer. Because of the diploid nature of mammalian cells, both alleles must be inactivated for a cell to completely lose the function of a tumor suppressor gene. Thus, it requires two genetic events to inactivate a tumor suppressor gene mutation, while only one genetic event is required to activate an oncogene. A subset of tumor suppressor genes controls the ability of the cell to maintain the integrity of its genome. Cells with a deficiency in these genes acquire an increased number of mutations throughout their genomes, including those in oncogenes and tumor suppressor genes. Many of these cancers appear to harbor a different kind of genetic instability affecting the loss or gains of whole chromosomes or large parts thereof, as explained in more detail below. Oncogenes in human cancer. Oncogenes are present in the genomes of retroviruses that are capable of causing cancers. The non-mutated cellular homologs, that is the counterparts, of these viral genes are called proto-oncogenes and are often targets of mutation or aberrant regulation in human cancer. Many oncogenes were discovered on the basis of their presence in retroviruses. Some other oncogenes, particularly those involved in translocations characteristic of particular leukemias and lymphomas, were identified through genomic approaches. Note, translocation. It means a change in location. It often refers to in genetics when parts of a chromosome is transferred to another chromosome. Some of the oncogenes previously found in retroviruses like ABL were involved in chronic myeloid leukemia (CML), whereas others were new like BCL2 involved in B-cell lymphoma. Note, this gene is a proto-oncogene that encodes a protein, tyrosine kinase, involved in a variety of cellular processes, including cell division, adhesion, differentiation, and response to stress. The activity of the protein is negatively regulated, that is, inhibited by its SH3 domain, whereby deletion of the region encoding this domain results in an oncogene. Note, domain, a specific physical region or amino acid sequence in a protein which is associated with a particular function or corresponding segment of DNA. In the normal cellular environment, proto-oncogenes have crucial roles in cell proliferation and differentiation. Oncogene examples: BRAF, function serine threonine kinase, alteration in cancer, point mutation, results in neoplasm, melanoma, thyroid cancer, colorectal cancer. Serine threonine kinases, STKs, play important roles in cellular functions such as growth, differentiation, and metabolism. A point mutation is a genetic mutation where a single nucleotide base is changed, inserted, or deleted from a DNA or RNA sequence of an organism's genome. Point mutations have a variety of effects on the downstream protein product. CCND1, function cell cycle progression, alteration in cancer, amplification, results in neoplasm, esophageal, head and neck cancers. Amplification, an increase in the number of copies of a gene. There may also be an increase in the RNA and protein made from that gene. Gene amplification is common in cancer cells. Some amplified genes may cause cancer cells to grow or become resistant to anti-cancer drugs. CTNNB1 function signal transduction. Alteration in cancer, point mutation. Results in neoplasms, colon, liver, uterus, adenomas. Note, signal transduction. Signal transduction is the process in which binding of an extracellular messenger to the surface receptor is translated into changes in biochemistry, cell biology, and gene transcription that make it possible for the cell to respond to the information that was received. IDH1 function chromatin modification. Alteration in cancer, point mutation. Results in neoplasms, glioma, a type of tumor in central nervous system. Note, chromatin modification. Chromatin remodeling is the dynamic modification of chromatin architecture to allow access of condensed genomic DNA to the regulatory transcription machinery proteins and thereby control gene expression. MDM4 function inhibitor of p53. Alteration in cancer, amplification. Results in neoplasms of breast. MYC function transcription factor. Alteration in cancer, amplification. Results in neoplasms, prostate, ovary, breast, liver, pancreas. CRAS function GTPase. Alteration in cancer, point mutation. Results in neoplasms, colorectal, lung, and pancreas. The normal growth and differentiation of cells is controlled by growth factors that bind to receptors on the surface of the cell. The signals generated by the membrane receptors are transmitted inside the cells through signaling cascades involving kinases, G proteins, and other regulatory proteins. Ultimately, these signals affect the activity of transcription factors in the nucleus, which regulate the expression of genes crucial in cell proliferation, cell differentiation, and cell death. Oncogene products function at critical steps in these signaling pathways. For details, see videos on cancer cell biology in Medicine for Anyone YouTube channel. Inappropriate activation of these pathways can lead to tumorigenesis. Mechanisms of oncogene activation. Point mutation, alternatively known as single nucleotide substitution, is a common mechanism of oncogene activation. For example, mutations in CRAS are present in more than 95% of pancreatic cancers and 40% of colon cancers. Activating CRAS mutations are less common in other cancer types, although they can occur at significant frequencies in leukemia, lung, and thyroid cancers. These mutations lead to activation of the mutant RAS protein. DNA amplification. The second mechanism for activation of oncogenes is DNA sequence amplification, leading to overexpression of the gene product. This increase in DNA copy number may cause recognizable chromosome aberrations. Numerous genes have been reported to be amplified in cancer. For example, NDM2, GLI1, CDK4, and TPS-PAN31 at chromosomal location 12q13 to 14 have been shown to be co-amplified in several types of sarcomas and other tumors. Note, each chromosome is divided into two sections, arms, based on the location of a narrowing, that is constriction, called the centromere. By convention, the shorter arm is called P and the longer arm is called Q. Amplification of a cellular gene is often a predictor of poor prognosis. For example, ERB2, HER2 is often amplified in aggressive breast cancers. NMYC is also often amplified in neuroblastoma. Chromosomal rearrangement. Chromosomal alterations provide important clues to the genetic changes in cancer. The chromosomal alterations in human solid tumors, such as carcinomas, are heterogeneous and complex and occur as a result of the frequent chromosomal instability observed in these tumors. In contrast, the chromosome alterations in myeloid and lymphoid tumors are often simple translocations, that is, reciprocal transfers of chromosome arms from one chromosome to another. Representative examples of recurring chromosome alterations in malignancy and the associated gene rearranged or deregulated by the chromosomal rearrangement. Examples: gene BCR-ABL in translocation 922 Q34 Q11. This translocation causes a new fusion protein. This fusion protein is made from pieces of two genes that get joined together to be produced. This fusion protein resulting is a potent cell death inhibitor. Malignancy. Example: chronic myeloid leukemia. Gene BCL2 18q21 and IGH 14q32. Translocation 1418 Q32 Q21. Malignancy: follicular lymphoma. Translocations are often observed in liquid tumors in general and are particularly common in lymphoid tumors, probably because these cell types have the capability to rearrange their DNA to generate antigen receptors. Indeed, antigen receptor genes are commonly involved in the translocations, implying that an imperfect regulation of receptor gene rearrangement may be involved in their pathogenesis. In addition to transcription factors and signal transduction molecules, translocation may result in the overexpression of cell cycle regulatory proteins or proteins such as cyclins and of proteins that regulate cell death. Recurrent translocations have more recently been identified in solid tumors such as prostate cancers. For example, fusions between TMP-RSS2 and ERG, which are normally located in tandem on chromosome 21, contribute to more than one-third of all prostate cancers. The first reproducible chromosome abnormality detected in human malignancy was the Philadelphia chromosome detected in CML. This cytogenic abnormality is generated by reciprocal translocation involving the ABL oncogene on chromosome 9, encoding a tyrosine kinase, being placed in proximity to the BCR gene on chromosome 22. The consequence of expression of the BCR-ABL gene product is the activation of signal transduction pathways leading to cell growth independent of normal external signals. So, dear viewers, this is the end of part 2 in the 3-part series on cancer genetics in the Medicine for Anyone YouTube channel. Thank you for watching the video.